In this video, we're going to demonstrate the dissection of a bovine eye. This video is meant to be a guide for your dissection in the lab, but in the lab, each group should be sure to dissect their own cow eye so that you can appreciate how the structures look and feel. You'll be expected to recognize these structures on a real eye during your practical exam. So first we're going to take a look at the external features of the eye, and first we'll look at the anterior features of the eye on the external surface. The most anterior feature that you see is this round, dark structure. And this is the cornea. And the cornea is where light enters the eye. And in a living cow, this is transparent. But when the eye is fixed, it becomes a little bit cloudy. So that's the cornea. Then the posterior 2/3 of the exterior eye is the sclera, or this is what we call the white of the eye. And this is the tough outer covering of the eye that confers the spherical shape to the eyeball. As we turn to the posterior aspect of the eye, most of what we see here is adipose tissue that cushions the eye in the eye socket. And you may be able to see some remnants of extrinsic eye muscles that move the eye. But the most important thing you're going to be looking for in the posterior of the eye is right here. And this is the optic nerve as it exits the eye. And that's the features of the external eye. So next we'll take a look at the interior of the eye. So you're going to start with puncturing the eye with the sharp end of your scissors. And be careful when you do that because the eye is filled with, might be filled with preservative fluid or other natural fluids that could spray out. And then you'll just make a frontal section that's going to divide the eye into anterior and posterior halves. And what we usually do is leave the anterior part of the eye attached at one point to the posterior. Okay, so there's your frontal section dividing the eye into anterior and posterior halves. So now we'll open up and look inside the eye. This jelly-like substance within the eye is the vitreous body. And you'll have to clear that out of the eye to see the internal structures. So you can see this sort of clear jelly-like substance. That's the vitreous humor. We'll move that out of the way. Okay, so we'll start on the anterior part of the eye. The first thing you're going to notice is this hard structure right here in the middle, and that's the lens. And typically students are sometimes surprised to see the texture of the lens. It's quite hard. It sort of looks like a flattened marble. So we'll move the lens over with the vitreous humor. After we remove the lens, you can see the cornea from the interior of the eye and the pupil, which is the opening that surrounds the cornea. And surrounding the pupil, if you look closely, you can see there are two rings of dark pigmented tissue. And the ring that directly surrounds the pupil is the iris that regulates the amount of light that comes into the pupil. Let's see if I can get a better shot of it. There's the iris. And then surrounding the iris is this ridged ring. And if I move this a little bit, you can see that this outer ring has ridges in it, almost like a bingo chip. And that's the ciliary body. So the inner dark ring is the iris, and the outer ridged ring is the ciliary body. Now we're looking at the posterior of the interior of the eye. And the first thing you'll see is this beige membrane here. And that's the retina that contains the photoreceptor cells of the eye. Since one of the functions of the vitreous humor is to hold the retina flush against the back of the eye, once we remove the vitreous humor, the retina tends to collapse when you're dissecting the eye. So you can see that it sort of crumbles up. Okay. You can also see that the retina is mostly detached from the eyeball, but it remains attached right here, and that's the point of exit of the optic nerve. The layer underneath the retina is the choroid layer, and in cows the choroid includes an iridescent reflecting surface right here. You can see this sort of shiny green layer, and that's called the tapetum lucidum. This is a specialized surface that reflects light within the eye, and it's found typically in animals that live under conditions of low intensity light. And you won't see this tapetum lucidum layer in human eyes. If you peel back the choroid a little bit, you can see that it's mostly a black pigmented layer. Gotta get underneath the choroid here. Okay. Let's see if we can show that a little bit better. Okay. And that's more typical of what you'll see in a human eye. The choroid contains melanocytes, which cause it to be pigmented and black like this. And if you look at this in cross-section, you can actually see that underneath the choroid layer is the cross-section of the sclera. So here we have the white of the eye again. See if I can show it a little better, right there. So in the internal eye, we have first the retina, then the choroid, then the sclera. And here's the sclera right here. And that's the end of our demo on the dissection of the bovine eye. You'll be able to see these structures more clearly on the real eyes in the lab, so be sure that you dissect an eye with your group in the lab this week. And if you need a little bit more guidance, please be sure to ask your lab instructor.